Protein structure prediction is the inference of three-dimensional structure of a protein from its amino acid sequence. That is, the prediction of its secondary and tertiary structure from primary structure is called protein structure prediction. Now let us understand what is the importance of protein structure prediction. It is uses protein structure prediction uses to fill the gap between known sequence and structures. The structure of a protein is determined by its amino acid sequence. This in turn determines the secondary, tertiary, and quaternary structures. The function of a protein, be it enzymatic, structural, signaling, is determined by its structure. Another important function of protein structure prediction is to alter function of a protein, that is protein engineering. Protein engineering involves synthesis of new proteins or amendment in the existing protein sequence or structure to achieve desired functions. Another purpose or importance of structural prediction is for rational drug design. One can also predict which molecules or drugs can efficiently bind and how they will bind to protein. This is of utmost importance for designing of drug. Structure prediction is done because it is very difficult and tedious to determine structure of a protein experimentally. Actually, the importance of structure prediction is structure that dictates function. That is, proteins are fairly large molecules with particularly complex structures that allow them to perform their functions. Without form or structure, there is no function. If the protein structure is known, then one can predict its function on the basis of structural similarity. One can also predict which molecules or drugs can efficiently bind and how they will bind to protein based on their structure. When we discuss the protein structure prediction, there are different levels of protein structures. Primary protein structure, secondary protein structure, tertiary protein structure, and quaternary protein structure. Complexity is low for primary protein structure, then the complexity increases to secondary, tertiary, and the complexity becomes so high with quaternary protein structure. As mentioned earlier, primary protein structure is a sequence of amino acids. Proteins consist of amino acids linked by peptide bonds. Secondary protein structures can be in alpha form, beta sheet forms, or coils or loops form. Most common structure is alpha helix. It is a local conformation primarily stabilized by hydrogen bonding. There is some intra-linked polypeptide bonds in the alpha helix shape. More complex structure of protein is tertiary protein structure. It is three-dimensional conformation. Tertiary protein structure consists of intra and interlinked polypeptide bonds. Several secondary protein structures together is formed in tertiary protein structure. Then the most complicated structure is quaternary protein structure, that is combination of multiple polypeptide chains are come together in quaternary protein structure. The structure prediction of quaternary protein structure is extremely difficult because of its high complexity. We are trying to predict the protein structure. Now, when we think about secondary structure prediction, the one purpose of secondary structure prediction is to predict the tertiary structure prediction. Then, the function of a protein can be predicted by knowing the secondary structure prediction. The types of proteins can be classified by knowing the secondary structure. Then the possibilities of structural change for a protein can be predicted when we know the secondary structure prediction information. The possibility of remote homology between proteins can be identified when we know the secondary structure. The detection and alignment of secondary structure prediction helps to identify the remote homology and between the proteins. Secondary structure prediction helps on detecting transmembrane regions, solvent accessible residues, and other important features of molecules. Then it also helps to detect hydrophobic region and hydrophilic region in the secondary structure of protein. Now, to predict the various protein structure, we should have a basic idea about the architecture of protein. Proteins consist of amino acids linked by peptide bonds. A chain of several amino acids is referred to as a peptide. Longer chains are often called polypeptides or proteins. When two amino acids are covalently joined, one of the amino acid loses a hydrogen H plus from its amine group, while other loses an oxygen and hydrogen OH minus from its carboxyl group, forming a carbonyl CO group and water. The result is a dipeptide, two amino acids joined by a peptide bond and a single water molecule. In a polypeptide, the amino acids are sometimes referred to as amino acid residues because some atoms of the original amino acid are lost as water in the formation of peptide bonds. Like DNA and RNA molecules, polypeptides have a specific directionality. The amino terminus or N terminus of the polypeptide has an unbounded amide group, while the carboxy terminus or C terminus ends in a carboxylic acid group instead of a carbonyl. Protein sequences are usually considered to start at the N terminus and progress toward the C terminus. The sequence of amino acids that comprise a protein completely determines its three-dimensional shape, its physical and chemical properties, and ultimately its biological function. This sequence is called the primary structure of the protein. Now, as you can see, the different amino acids, glycine, alanine, etc. It is worthwhile to learn one-letter code for each of these amino acids. There are 20 amino acids. These codes allow protein sequences to be expressed as a string of characters, much like nucleotide sequences. And these 20 amino acids can vary in their shape, in their size, in their polarity, in their hydrophilic nature, hydrophobic nature, etc. etc. Now let us understand more details about the secondary structure of protein structure prediction. The non-side chain atoms of the amino acids in a polypeptide chain form the protein backbone. The bond lengths and planar bond angles of the covalent bonds in the backbone are more or less fixed. In other words, the peptide group is rigid and planar. The two bonds between the alpha carbon C alpha and the other backbone atoms are the only two rotatable bonds in the protein backbone. All flexibility in the protein backbone is derived from the rotation of these two bonds. The angle of rotation about the bond between the amide nitrogen and alpha carbon is referred to as phi, while the angle of rotation about the bond between the alpha carbon and the carbonyl carbon is called psi. The backbone conformation of an entire protein can be specified in terms of the phi and psi angles of each amino acid, as shown in this figure. Not all values of phi and psi are physically realizable. Some phi or psi combinations result in a steric collision, that is, physical overlap of the space occupied by atoms between the side chain atoms of one residue and the backbone of the next. Remember, in the protein structure prediction, or when we learn protein secondary structures, alpha helix is the most abundant secondary structure. Around 3.6 amino acids exist per turn, and hydrogen bond is formed between every fourth resid, and the average length is around 10 amino acids or 3 turns, and it varies from 5 to 40 amino acids. Another pictorial representation of protein secondary structure with these beta-pleated sheets, alpha helix, random coil, etc. are shown in this figure. So, based on CN bond phi and CC bond psi, let us understand or remember the protein secondary structures of alpha helices, beta sheets, and various coils and loops formed, as shown in this figure. The Ramachandran plot illustrates the allowable values for phi and psi for non-glycine residues. Because of their lack of a side chain, glycine residues have a much greater range of allowable phi and psi angles than most other residues. So this Ramachandran plot shows the frequency of phi and psi observed for residues in folded proteins. Whenever we make the predictions of secondary structure, accuracy we have to discuss. There is a big problem sometimes. Secondary structure prediction algorithms use a variety of computational techniques, including neural networks, discrete state models, hidden Markov models, nearest neighbor classification, and evolutionary computation. Most current secondary structure prediction algorithms begin with a set of aligned sequences produced with algorithms such as BLAST, FASTA, and CLUSTAL-W. Using these aligned sequences, the degree of conservation of each of the amino acids in the target sequence is calculated with a protein sequence and the corresponding conservation level as input. Modern secondary structural prediction algorithms such as PhD and Predator can obtain accuracies in the range of 70 to 75%. Output from a secondary structural prediction algorithm is generally similar as shown here. Let the sequence be MRIILLGAPGAGK, etc., etc., etc. Then the output will be like dash 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 H H H H H H H, etc., dash H H, then some small h h h dash, then capital E, small e dash dash, etc. Here the capital H and small h represents the predictions of helical conformation, where capital H is with strong conformation and small h represents the confidence level as weak. Similarly, E and small e represents the predictions of extended beta strand conformation. Capital E represents strong confidence, whereas small e is the prediction with weak confidence.